Muscle fibers originate from stem cells known as myoblasts. They fuse to form each muscle fiber early during the development. And some of those cells, known as satellite cells, that are unspecialized myoblasts, remain embedded in the muscle fiber and endomysium. And these satellite cells play a very important role in regeneration of damaged skeletal muscle tissue. And the other part of muscle fiber is the mitochondria. So mitochondria is the organelle that synthesizes ATP, which is a form of energy the cells can use. So mitochondria is an organelle that is packed very early into the spaces between myofibrils during development. Now, the next germ you should be familiar with is sarcolemma. Sarcolemma is the term for plasma membrane of muscle fiber, and the infoldings of sarcolemma are known as transverse tubule that we'll look at in just a minute. Now, the muscle fibers are multinucleated because they fuse to form muscle fibers during early stages of development, and they are pressed against the margins of the sarcolemma, which is the plasma membrane of the muscle fiber. And the cytoplasm of the muscle is known as sarcoplasm. The sarcoplasm consists of myofibrils, which are long protein cords that occupy most of the sarcoplasm. So the next important component of sarcoplasm is glycogen. Now, glycogen is the carbohydrate stored to provide energy during exercise, and myoglobin is the red pigment that provides oxygen needed for muscle activity. So this picture over here shows a muscle fiber, and if you peel off the sarcoplasmic reticulum, you see long cylindrical myofibrils. So these are the myofibrils. And these myofibrils contain thick and thin filaments, both together, known as myofilaments. So the thick and thin filaments together are known as myofilament. The thick filament is made of actin, the thick filaments are made up of hundreds of myosin molecules, and the thin filament is made up of actin, tropomyosin, and troponin. The muscle fibers also contain elastic myofilaments, known as titin, that are not visible over here. Now, if you look at these blue structures in the muscle fiber, these are known as the sarcoplasmic reticulum. The sarcoplasmic reticulum are the smooth endoplasmic reticulum that form a network around each myofibril, and at the end of the sarcoplasmic reticulum, you see these blue structures that look like tires. Now, these are the dilated end sacs of sarcoplasmic reticulum, which cross the muscle fiber from one side to the other. And these disc-like sacs are known as terminal cisternae. Now, the terminal cisternae act as a calcium reservoir, and it releases calcium to activate contraction. Now, sandwiched between the terminal cisternae, there is another structure known as a transverse tubule. So this purple structure over here is known as transverse tubule, and the transverse tubule, their main function is to conduct impulses from the surface of the cell, which is the sarcolemma, down to the other structures of the cell. So the terminal cisternae and transverse tubule together is called a triad. So the myofilaments are made up of thick filaments and thin filaments, and also elastic filaments. So in the myofilaments, the thick filaments are made up of hundreds of molecules of myosin, and thin filaments are made up mainly of actin and tropomyosin and troponin. The elastic filaments are majorly made up of titin, and the elastic filaments basically anchor thick filaments to the Z disc and the M line, which is the middle region supporting proteins that hold thick and thin filaments together. So we look at each of these filaments in a lot more detail. So this shows the side view of a myofibril, and you can see that there are a bunch of myofibrils in a muscle fiber or a muscle cell. If you peel off the outer layer, you see a bundle of thousands of myofibrils. Now, so this Z disc or Z line over here are the striations we see under the microscope, along with these other protein filaments that provide the anatomy that you see under the microscope. So anchored to our Z lines, we have actin filaments, and in this region known as the A band, you have overlapping filaments of actin and myosin. Now, from one Z disc to another, it is called a sarcomere. So sarcomere is the space between two Z discs, and the sarcomere is the functional unit of muscle. So the muscle fiber, each muscle fiber is made up of thousands of sarcomere. Now, in between the sarcomere, you have titin, which is the elastic filament, and then you have these red filaments over here that are the myosin filaments, and the blue filaments are representative of actin filaments. So this picture over here shows alternating dark and light bands that you would see under the microscope, and here these dark lines are the Z discs. So one Z disc to another is one sarcomere, and below this you see an artist's rendition of a structure of a sarcomere. So each myofibril consists of thousands of sarcomere. So the Z disc separate one sarcomere from the next, and in this region you have actin filaments and the titin filaments. So the green ones are the titin filaments, that are the elastic filaments. You don't see any myosin filaments in the Z band, the Z disc. Then you have the A band. So A band is the dark middle part of the sarcomere, and in this region you have thick and thin filaments overlapping each other. Thick filaments are the myosin filaments, and the thin filaments are the actin filaments. Now the I band, keep in mind, does not have any myosin filaments, but the A band has overlapping actin and myosin filaments. Then you have the H band or the H zone in the center. Now the center of each A band, which contains thick filaments but no thin filaments in this region, is called the A zone, and the M line is the center of the sarcomere, which contains supporting proteins that hold thick filaments together. So this region is made up of supporting proteins that hold the thick filaments together. So right over here, you see a portion of sarcomere that show a region where thick and thin filaments overlap. And you can see that the thick filaments are made up of hundreds of little structures in here. And each of these structure which has a head is a myosin molecule. If you look at the structure of thick filament, you see that the thick filament is made up of several hundred myosin molecules, and each molecule is shaped like a golf club. And each molecule contains two chains that intertwine to form a shaft-like tail, and it has a double globular head that pulls against the actin filament to allow muscle contraction. Now the heads, when they are bundled together, are directed outward in a helical manner around the thick filament bundle. And if you look at the overlapping region of a sarcomere, there's a region where you don't see any globular heads, nor do you see actin. So this region is known as the bare zone. So the bare zone does not have any globular heads in the middle. The thin filament is made up of tropomyosin molecules and troponin complex. Now the tropomyosin molecule consists of these bead-like structures, about 6 or 7 of these bead-like structures known as G-actin subunits. Now G stands for globular actin subunits that can bind to the head of myosin molecules. Now the troponin molecule is a small calcium binding protein on each tropomyosin molecule. So the yellow structure is the troponin complex. Now tropomyosin and troponin work together to prevent actin from binding to anything, and it helps a lot in the contraction process. So this picture again shows alternating dark and light regions of the striated skeletal muscle. And the one thing we didn't talk about is the elastic filament, which are made up of titin proteins. So these titin proteins are springy proteins and run through the core of the thin filaments. So they basically anchor the thick filament to the Z disc over here. Okay, and the role of elastic filament is to prevent overstretching and to allow recoiling of the muscles. So this Z disc is the end of the actin filament where both actin and titin filaments come together. So the H band, you don't see any globular heads. So this region is called the bare region. And the A band is the region where the thick and thin filaments overlap. The M line is a line of accessory proteins that hold the myosin together.